This protocol is designed to determine the submitochondrial localization of yeast mitochondrial proteins, which is considered as a fundamental step during mitochondrial protein function elucidation. The protocol is suitable for all yeast strains maintained in different growth conditions, representing a powerful tool for researchers studying mitochondria.
To begin with, streak cells from a glycerol stock stored at -80 degrees Celsius onto a YPD agar plate to isolate single colonies from the strain of interest, and incubate the plate at 30 degrees Celsius for 2 to 3 days. Prepare a starter culture by inoculating 2 to 3 individual colonies from YPD agar plate in a 100 milliliter Erlenmeyer flask containing 10 to 30 milliliters of YP gal medium. Incubate the flask at 30 degrees Celsius for 24 hours with vigorous shaking at 180 to 200 rotations per minute. Dilute the starter culture into 1 liter of fresh YP gal medium to an optical density less than 0.1 at 600 nanometers. Cultivate the cells at 30 degrees Celsius with vigorous shaking at 180 to 200 rotations per minute for about 12 hours until optical density reaches 1 to 1.5. Perform the isolation of highly purified mitochondria as described in the text, and determine the protein concentration of the highly purified mitochondrial preparation using the Bradford assay following the manufacturer's instructions. Adjust the protein concentration to 10 milligrams per milliliter with ice.